So guys, today we are going to be speaking about the breeding innovation for tomorrow's market. And we have the chance of having four amazing panelists. We have Atia Ferrouz, Jacob Tott, Sylvain Gaudreau, Max Jones, and we're missing one. Yeah, Max Jones is not here, so Jack Munns, Dr. Jasmine Munns is there also. I'll let them introduce themselves and their specialty, and then we'll dive into the subject.
Oh, hi there. My name is Atia Ferrouz. I'm the CEO and founder of AgCan Consultancy. I've been in the cannabis space since 2018 in Canada, but I'm currently based in Australia and Asia Pacific. I specialize in GACP and GMP compliance and also tissue culture. I own a tissue culture, genetic banking, and hoplet and viroid testing lab in Thailand.
Good morning. I'm Jack Munns. I am the research director at 3R Biotech. We're a Vancouver-based ag biotech company. We do affordable tissue culture and hop latent remediation, hop latent testing, among other pathogens, focusing on TC for storage, germplasm, and HLVD remediation. But our key comments are sort of affordability and quality.
Hi, everybody. My name is Jacob Toth. I have 10 years of experience in breeding professionally. I have a PhD in cannabis breeding from Cornell, and I currently manage the plant science department, or Gannogram Global, the largest cannabis company in Canada by market share, as we're proud to say. In this plant science department, we do cultivation improvement, we do post-harvest research, we do breeding, and we do biotech.
My name is Sylvain Gaudreau. I'm the head researcher for CANNA. We specialized in research that has to do with cultivation aspects. Our latest paper we wrote is called The Forgotten Half. It basically talks about how we put aside male plants in this industry.
Thank you very much. So, cannabis in itself has been bred for thousands of years, but not at the commercial level that we are seeing now. So many questions are coming up with what is the future for cannabis propagation? Is it tissue culture? Is it F1 autoflowering seed? And so on. So today we're going to dive into two main questions. The first core question will be on the seed-based production, F1 autoflowering genetics. And then we will look into, if it's not F1 autoflowering, what should breeder prioritize? Tissue culture, male plants, what is important? So let's first dive into the F1. I would love for you guys to talk to me about what you know about those F1 seeds, what's the opinion, what's the future for them. We can start by, yes?
So F1 seeds, obviously it's very, very common in other big ag. Corn being a prime example. Starting from a seed stock is great in theory to have consistency. One of the problems right now, I think, would be pure breeding lines take a long time to develop. And stable purebred lines, when you match them with another purebred line, aren't always going to show hybrid vigor, so finding the right matches. So each F1 seed, you're taking anywhere from 8 years to 10 years to develop one of the parents as a purebred line that's stable, along with the other parent. So it's going to be a while before we have any real diversity among all those. It's a combinatorial factor. So it's like if you have one pair, that's one type of seed you're going to get. And everyone after that, it's like three. So you have three different parents. You can make, 6 combinations. And it gets a little bit bigger each time, but it's going to take a while to actually get diversity. And even then, you're selecting for things you don't even know if you're going to want 5 years from now, right? So we're talking about things like CBG, HEHV. 5 years ago, did we know that that's where things were going to go? And so big picture, I think tissue culture and gene banking, synthetic seed, cryopreservation are going to be essential to preserving genetic diversity so that you can harness it for F1 seeds later. And F1 seeds is very much a big picture play. So I think it's a tool. Eventually, maybe it could be the full picture, but it's still you're going to need a gene bank somewhere to keep the diversity that we have from thousands of years of breeding. You don't want to lose that diversity. You can't get it back. So gene banking and germplasm storage is going to be essential to it.
Thank you. Yes, go for it, Jacob.
I feel I have very similar opinions. I think the main drawback is the speed at which we can get these inbred lines. It takes a really long time to develop these inbred lines that will be the parents. Compare that to our clonal breeding program. We can just do a cross, do our selections out of that. If we want to make our own F1 seeds, we need to inbreed and inbreed and inbreed and inbreed, and then we do the cross. And who knows what is going to be the market 5 years down the line, or however long it's going to take. Here at Organogram, we do grow a lot of F1 seeds that we're largely sourcing third-party right now, and they grow pretty well. If you look at the cost per gram, we can't beat the Autoflower F1 seeds for cost per gram. The current stuff that some of the best companies are coming out with right now are competitive in terms of elite aromas, high THC, high yields as to some of our other common clones. But just sort of in terms of what comes next, it's just really slow to do the breeding part of it.
I think that when we talk about like what cannabis looks like, all of us really look at how the industry is right now, right? Because that's what we're seeing, and that's how it's kind of been for the past, let's say, 8 years in Canada, which is very new. And so in a very new market, especially one that I would say is like so heavily consumer dependent, they're the high THC pushers, they're the ones who want these specific flavors. Things are popular in California. Next thing you know, we need them here in Canada. So we're in a very consumer driven market right now, and we're in a very highly recreational consumer driven market right now. And as markets begin to become more medical, and we see markets like France that are coming on just for extracts, and the Philippines that's coming on just for extracts, this is where very high volume biomass becomes very important. And in fields, like we've had people in Thailand come to us and be like, I need some clones. I'm like, okay, how many do you need? They're like 300,000. I'm like, I'm not cutting you 300,000 tissue culture clones. Like, it doesn't make any sense. You have to look at getting a seed option, because for transport, for stability, for having that product at a very, very high volume over very large fields, a lot of our clients are massive producers outdoors in Zimbabwe and Democratic Republic of Congo, et cetera. And those are clients that I have for GACP certification and other services. And so I think when we look at who's going to be using seeds in the future, these very large-scale outdoor breeders are going to have, or outdoor growers are going to have to use seeds. It doesn't really make sense for them to develop the infrastructure to essentially have enough space for, typically they do a greenhouse or an indoor on-site, to do enough mothers and clones to cut once for the season, and then they leave that empty for the entire year, and then they refill it again. So I think for our clients that we work with who are indoor producers, I think that right now in the cannabis industry, with the level of instability in genetic preference and the fact that it changes like flavors of the month, I obviously run a tissue culture service, and we do that where we bank genetics for people. But I think ultimately long term, just as we see in other agriculture, when we're looking at replicating things around the globe, let's say you also own a company in 5 different continents, seeds are going to be the easiest thing for you to transport on all 5 continents to allow for consistency. So I see tissue culture having a really big place right now in the market, and I see us switching to seeds over time for if you're an outdoor cultivator producing a large biomass, or if you're a cultivator operating on multi-continent.
Are you concerned about outdoor grow needing a very specific type of seed? Because it's going to be a different... obviously hemp growers use seeds a lot. It's basically what they're using right now. But getting to a place where you're getting high biomass CSE cultivars outdoors, is there a concern with how long does it take to get seeds unique to that?
Yeah, for sure. And that's why I think it's like in the next 5 or 10 years we'll see that. And even when I talk to people, like I used to sell genetics when I worked in Canada like 6 or 8 years ago, and we see a lot of nurseries and breeders now. And when I'm trying to buy things from them, because I don't do that myself that much anymore, they just give me 1 C of A, and I'm like, what is this for? And they're like, it's for an indoor. And I'm like, when I ran genetics, I gave them to an indoor grower, an outdoor grower, and a greenhouse grower, at least 2-3 batches, pulled all of the data from that, and I would not feel comfortable selling a genetic unless it had data from a similar grow environment. And so I think we're very heavily focused on indoors because they're typically the cultivators that have more money. They can spend more on genetics. They have more of that budget for it. But I think as you're developing genetics, if you're a breeder, if this is a focus of yours, and if you want to serve multiple markets across multiple continents, it's really important to partner with growers and get that data.
What about synthetic seeds? So we can do synthetic seeds and ship those similar. TC still needs to be receiving on the receiving end, so you need a TC lab, but things like bioreactors can scale up TC fast. So the numbers of thousands, many thousands of plants is possible. I definitely agree that we're going to seeds, it's just going to take time. But when you have specific environmental variables you got to take into account as well, it might take even longer. So in the meantime, things like bioreactors to help scale TC into production runs is something that we're looking into. That really accelerate how you can scale TC. The big drawback for bioreactors is contamination, but if you do it on, we do it on modular small scale bioreactors like we link and sequence. So that way, if you lose one, you don't lose the whole crop. But as far as like the shipping compliance goes, there are other ways besides seed to go international. And tissue culture basically is already phytosanitary, so it makes it kind of easy to get cross borders that way.
Would love to hear Sylvain. What's
Well, seeds for me are definitely the future. It's the easiest way to breathe back in what we breathed out over the last 10 years. Unfortunately, by chasing that high THC point, we've breathed out a lot of qualities that these seeds need to have, especially when we go around world markets, that we need strains that are heat resistant, frost resistant. We had one that we produced, like it can hold up to 5-6 frosts easily. And those are traits that we need to breathe back in very, very importantly.
Okay, well, you all seem, I think we can all agree there is a right case of usage for autoflowering seeds, but I would be curious if you guys have maybe stats that can be shared. So let's say autoflowering seeds in Canada, we have Royal Queen Seed that I think is trying to pump some out. We have Phylos that are pumping some out. Do we have some germination rate on those?
We partner very closely with Phylos, and the germination rate has been fine, and the quality of at least some of the hybrids that we've been growing have been sufficiently good that we've been growing them extensively indoors, and we are also partnering with various people all across Canada to grow them outdoors as well.
Okay, and when you say, sorry, I like numbers, if you're comfortable, what is fine?
I will say above 90%. I don't want to say the exact
Yeah, no,
numbers,
fair,
and
that's
it's
fair.
going to be variable depending on a lot of things, but we're seeing above 90% generally.
And the process from germinating the seed, putting it in, let's say, a Rockwell cube or cocoa or whichever the growing medium, and getting it through those initial 2 weeks, did you also had a lot of success versus doing clone?
Yeah, so it was a little bit of a learning curve growing seeds at scale compared to the clonal propagation that we were doing, and it took a little bit of optimization, but we were able to get them just as good as our clonal success rate, the seeds. So some things needed to change. The seeds have a very thirsty taproot early on compared to the clones, so our parameters are different, but with a little bit of optimization, we got them just as good.
How long does it extend your growing cycle?
It's actually quite a bit shorter.
Okay. And do you see a lot of variation? Like, let's say you run 2,000 seed, do you see 2-3 phenos? Or?
Yeah, so that's going to depend a bit on the genetic variability of the mother plants for those, and that's just sort of dependent on the work that our collaborators at Phylos or other companies have done. So that's quite variable. The ones that we grow commercially, they're quite good. We see them, they start to flower the same. We get the same yield. We get the same aroma. We get the same potency across all plants. There's certainly more variation than we see in the clones. But one thing that, we talk a lot about internally is that even within a given clonal plant, like the tops are 2 percentage points higher THC than the bottoms. So if I'm growing autoflowers and one's 2 percentage points different than the other, oftentimes we see similar amounts of intra-plant as inter-plant variations comparing the clones and the seeds.
Very nice.
For us, I have the pleasure of working with Dustin McLean with the seeds and everything, and so far all the seeds we've tested run between 92 and 98% germination rate. So for us, that's a big win right there.
Okay.
And we're playing around also with, instead of using seeds, we're trying to use germs. So you let them pop out of the seed and replant them, and we're having a little bit of a success, but it's a hard process that it wouldn't be viable for an industry, but for research, it cuts corners really nicely.
Okay, and so we can dive maybe a little bit more into the second part of this panel, which is, if not F1, then what? Do we go tissue culture? Do we invest maybe in males to start our own breeding line? If, the nature of our business is we're CPGs, right? We need to be able to pivot quickly. One of the things I think a lot of our producers are afraid is there's only so many autoflowering supplier. So do I want to go up against Organigram when they're already producing those strains that I can buy from Phylos? So what would be the alternative for growers?
Yeah, I think when we look at the current F1 seeds provider, it's very limited, right? So like I think I have like 6 clients who also use Phylos in Canada. Like so then you're like getting to the problem that you had, back in the day when we had tissue culture providers that only had like 6 genetics. I think what was it like there was a cherry genetic that everyone had at one point. It was like ridiculous. Like every single grower had the same stuff. So that is a challenge for sure. Everybody wants variability. They want to have something special. They want to have something unique, and you can only breed so fast. So I mean, how many new genetics can Phylos put out in 1 year? When it comes to starting your own breeding program, I would say that is a very ambitious endeavor. I don't usually recommend a lot of people do it. I think a lot of people think it's going to be a lot easier than it is. I think if you're also cultivating on the same site, you have to worry about completely different concerns than if you're just a breeding only site. And so I think that I always tell people what I find in the cannabis industry, because it's new for sure, is that everyone's always trying to suck up all the supply chain into themselves. And generally speaking, across most other businesses, that's not a good idea. It's really important to find good partners. Of course, if there's a gap and you can't find anybody and you have to figure it out yourself, that happens in emerging markets. But for the most part, if you're really heavily invested in using seeds for some reason, then you can start talking to these providers now. And you can actually maybe even discuss putting funding towards developing your own lines with them, but not really having the whole responsibility unto yourself. And then, of course, in the short term, many people are doing tissue culture. What I always tell people when growers are like, why should we do TC? I say, for me, it's like pressing Control Save on your work, Some of the breeders come to us and they've like had genetics for 10, 15 years. They've been keeping them as moms in the back of a grow room, just recutting them every 3 months over and over. And then that leads to obviously stressors coming into the mother and affecting genetics, like epigenetic stress. So what I always say is like, if you've bred something and you have an active plant of it, we will basically press Control Save for you and we'll hold that. And we can, of course, we usually resupply back fresh mothers every 3 moms for our clients, but of course we can bank them for decades if needed. And that's what I think is right now, in my opinion, the best solution for most growers in the short term is tissue culture. And I think that will be the case for at least the next decade before we really start to see F1 be much bigger.
In-house tissue culture, should they contract a lab? Do you think that's an expertise that, let's say, an LP, a certain size, should acquire and control themselves?
I think so. And so we have this larger LP, like one of the biggest LPs in Canada, approaching us right now because they are using a third-party tissue culture provider, and they're getting very nervous about that tissue culture provider's ability to keep running. And that's a very big risk, right? So we work with a lot of smaller LPs in Thailand because there are just not many that are that large, and so it makes no sense for them to develop. If anyone shows me the Athena tissue culture kit again and tells me they're going to bank their own genetics, I'm going to lose my mind. It is a waste of money. I will tell them that to their faces.
I succeeded twice with it.
You can do it for like 1 or 2 calls. I have people being like, I have 78 genetics. I'm going to bank it with this kit. I'm like, this thing does not look like it's going to provide a...
It is not professional.
No,
We agree.
and
it's a proof of
if
concept.
it's in your house or your backyard and you're not a commercial grower, it's totally fine. But we've had so many people buy this and then kill all their genetics and then realize that they can't continue on with it. So I would say, like, if you're quite small scale, it would make sense to use a third-party provider. I think as you get larger, as you develop larger genetics, I'm not saying you have to bank yourself, but having in-house capability of just your own hood and very basic skills that if that lab closes down, because if I close down my lab, I have to return plants back to them, right? They need to do something with them. So as if you're like a paranoid person or you feel like that's, that company is not viable long-term, then yes, having some sort of in-house capability, whether or not that means you're banking all 80 of your genetics yourself or if you're just having it there in case of emergencies, that's up to you.
She has a will of steel. I am getting distracted by this. She just kept on going, no flinching. Holy shit.
So we have 300 genetics in a 500 square foot space. So we can do it for very affordable. For a couple $1,000 a month, we can license you technology, low overhead. So we build our tissue culture hoods ourselves. The racks, we ship you media, so you don't have to make an autoclave is a huge expense. So we freight media monthly. So we have really cost-effective solutions for storing your own genetics if you have that scale. But we also have that ability to take on smaller in-house things. As far as like pheno hunting, synthetic seeds are great for this. You can put things into synthetic seeds and not touch them for 6 months, bring them back out when you have the handful that you want to keep and carry forward. So there's a lot of options available for that. As far as the breeding goes, I think differentiation, if somebody's brave enough to step away from the high THC and start trying to focus on some of the lesser known cannabinoids, things like that. There's over 100 cannabinoids. Yeah, we don't know which one's going to be important 5 years from now, but I think it's time to start, like, the people who differentiate themselves in that market. As far as breeding goes, that's something that makes you different. So I think that's as far as breeding and trying to have unique traits is like field, things that are ready to go outside versus things that are male traits, to try and keep some of that genetic diversity. If you have that, somebody else doesn't, that's a value add. So it's going to take space, right? Tissue culture can help minimize that space so we can store those things while you have a little bit of time, a little bit of space while you're doing your cultivations to actually work on that kind of thing. So cost-effective tissue culture is the only way you're going to store that many genetics to do those kind of bold moves into something that's not just THC, high CBD kind of strains. So.
Thanks. So to go back to the question about alternatives to F1 autoflower seeds, one thing that we've sort of touched on that I think is worth more discussion is outdoor grow compared to indoor grow. So what's nice about autoflowers is that they don't care about the photoperiod, so you can grow them anywhere. You can grow them in Thailand. You can grow them in the Arctic Circle if you really wanted to. You can grow them indoors, greenhouse, all that stuff. But non-autoflowers are often photoperiod sensitive, so they're going to flower at different times depending on what latitude you grow them at. And what we've seen, I've grown plants across Canada and across the States at very different latitudes. If you get the right photoperiod plant, they can way out yield the autoflowers because they just use the whole season. So like here, in Canada, you need something that's going to be fairly early flowering. What we've seen, we're quite successful with, is you grow the autoflower heterozygotes. Those tend to be a few weeks earlier, shorter critical photoperiods, start flowering sooner. So we get a lot more yield out of those compared to the true autoflowers outdoor because they just use the whole season. And then moving further south, you want longer, and you have more and more photoperiod to play with. But then once you get to the equator again, the story changes again. I don't have any experience growing near the equator, but I'd imagine there'd be some more interesting photoperiod things to think about there. And I guess just one other thing that I think is relevant to the question of how can we get either F1 autoflower seeds or just F1 seeds quicker. So the traditional process to get perfectly stable seeds is inbreed. But a lot of other crops use other technologies, notably like double haploids, where you can go straight to S infinity, totally homozygous at every locus in one generation. There have been people working on that in cannabis, but I'd really like to see more research in that area.
We actually have an expert there, so we'd love to hear you more talk about your male research, also the importance of a male. I think in autoflowering, a lot of us, what we experienced in the previous year is everything tasted the same because we all know it kind of came from the same breeding and the same crews, and everybody passed around the same plant. So yeah,
that's one of the main problems right now. We're creating this massive genetic bottleneck that you can trace back pretty much every cultivar we have back to like 12 to 15 major strains, and we just keep on playing around. So we're breathing out excessively important stuff out of the plants. Right now we're breeding stuff, and like any technique that you decide to go on your breeding future in your company is, I would say, go with your skill set. I have no preference to one to the other. What I think it's important is we need to start putting down on paper what we do, and we need to start evaluating the male plants also. Node spacing, pollen shed, pollen release timing, those are all things that could give you an edge and make those cultivars different from one to another. Right now, what we're working on is cultivars that are for outdoors that resist heat and frost. Most of the cultivars that we have right now that are grown in greenhouses and indoors, I would take outside and give them about a week and a half before it passes. We've bred it out, most of the resistance, to pursue a number that, for me, that's a number that means nothing whatsoever. Because I have, and pretty much here, everyone here can say the same, that you had THC up to 25%, that you went, and you had THC that was 15% that knocked you on your ass. And people goes, well, that's not possible. Yeah, actually it is. It's the way people react to different cannabis strains and everything. So those are important things. And we need to step a little bit back away from the fact that this industry is being led by the customer and not by us. And we should be the people educating the customers on where this industry is going, not the opposite way.
Perfect. And so for the last lightning round, I would like you guys to tell the crowd what would be maybe something you think, if you had an unlimited amount of money or you had no constraint, what would be your future propagation setup? Would you invest more in auto-flowering seed? Would you invest maybe a little bit of space in hunting a male? Would you find partners to do TC and breeding? What would be your best outcome for the next 5 years?
Yeah, so I mean, as I said, we have a tissue culture location in Thailand and opening up a second location in Australia. My plan is actually to have one on every continent. And the ultimate goal is essentially to set up somewhere central a kind of individual like research lab. So what I find like when people run tissue culture is they tend to try to do research at the same time. And it's like you're competing with two different things. You're commercially providing services, you're trying to do research. So I think there's so much interesting genetics research to be done in cannabis industry. And I think like what my goal is to have multi-locations on different continents, a single centralized research lab, and the ability for us to move products between continents. So when we look at bringing things into Australia, it's a 3-month quarantine. So every clone you send down there sits in a warehouse for 3 months. The staff are taking care of it. They look disgusting when they come out the other side. Oh my God. I'm just like, have you ever seen a plant in your life? Like, what did you do here? So that's the current process, and that's why I'm setting up my own tissue culture labs. I can go through that really rough quarantine period, get some good cuts off of that, and then reestablish better genetic lines. But the idea is to, each market has such different needs on what's preferential for genetics. Like, yes, high THC is very popular, but again, if you're serving a market where it's mostly outdoor grows, what's going to sell there is not the same as what's selling in an indoor grow market, right? So I think the globalization of the cannabis industry, to me, is like the most fascinating thing about the industry. And as you know, I've worked globally for quite a long time. And I think also this kind of interplay of genetics. So there's actually someone I'm talking to here at the show who has a bunch of genetics from like a really old like land race strains from a country in Africa, and he wants to work together to bank those. And I could then take those TC, send them over to Thailand, and crossbreed them there with like land race strains from Thailand. So I'm quite keen to do that in the next like 3 to 5 years. And I think that I'm a big firm believer that high THC is really not good for you in the long term, and it's not what's going to serve the market best. And it's also a really aggressive starting point to get people in and be like, I had this edible and it was like 50 milligrams. I had to go to the hospital. Like, it's not, not the best. Having a too strong edible is one of the worst feelings in the world, I would say, by far. And so I think that, being able to kind of go back to these more land race strains, which are obviously much lower potency, just really enjoy them for what they are, maybe do some crossbreeding to bring out some interesting characteristics, but not like driving towards this 38% THC point. I think that's going to be kind of what is really popular in about 5-10 years from now. And also like what's better for the industry is having these more like natural products.
For me, it's affordable cryopreservation and genetic banking like that. So these strains have been around a long time. So for seeds that are really old, it's somatic embryogenesis to bring them back into TC. So if they're not germinating, ways to get those, harvest the embryos out and grow them back into TC that way. But for the things that we have, you tell me what's important and make affordable cryopreservation, and I'll bank it for cheap. And cryopreservation, the important part is it stops the molecular clock. These things are aging every time you cut them and plant them. They're getting older and older and older until they're not the same anymore. So banking the genetics now so that we have them later when we figure out what it is we want in the future. Affordable cryopreservation is for me in the next 5 years. In the meantime, synthetic seeds slows the clock significantly. I mean, we're talking about instead of tissue culture every 3 weeks, it's tissue culture every 6 months. So that slows it down substantially, 6x or so. But really the long-term goal for me at 5 years is cryopreservation, which is what all the big, crops have been doing for decades. It's not new. It's just making it affordable so that you can tell me what's important because, you're the ones that know. So
I have 3, but I'm going to be quick. First, pie in the sky idea. If I had unlimited money, unlimited time, and no obligations, double tabloids all the way. A little more realistically, I'd be very curious as to sort of optimizing Photoperiod, figuring out that the genetic basis and being able to breed lines for every latitude, see what can grow best in every location. But more realistically, and this is something that we're working towards and have made some good progress lately, is sort of just developing a good idiotype, like what do we want our plant to be? We want it to be powdery mildew, pathogen resistant, we want diverse aromas. So figuring out the genetic basis of that and bringing that into the elite Autoflower F1s is really what I see in the next few years for us.
For me, it would be like a national registry for cannabis plants. Most crops in Canada do already have a registry. that you could look up traits and everything, all the information on plants. We don't have that for cannabis, and there's a few countries that already do this. We need to have a registry for male and female plants to be able to eventually, I know a lot of people are into the IA thing now and then, but I've seen that people use projection with IA to see what crosses will look like down the line, and having a national registry would just help a lot with doing this.
Very nice. So overall, what I'm hearing is a lot of agility. None of you would lock their self in. You need to promote agility because, again, if we can't evolve and we can't move with the market, we're not going to survive. So also start a tissue culture lab once you get to a certain size. Call it TIA. Is there any question in the audience for our wonderful panelists? Martin, you look like you have a question. No, yeah, you do. Come on. Mr. Bergeron in the back there, you look like you have a question. Everybody's too shy to talk to you guys. Oh, sorry.
Yeah, so the question is, comparing our seed crop cycle to our clone crop cycle, our seed crop cycle is shorter. Yeah, from taking the cutting or planting the seed, our crop cycle is faster from planting the seed.
Is that...
And that ends up saving us a lot of money. We get it costs us to grow a plant, so if we can grow them for fewer days, that's cost savings for us.
Is that due to root development from cuttings, or?
Well, yeah, that's a good question as to exactly why. It might have something to do with the roots. Might be something about how the autoflowers, we can grow them at a longer photoperiod, so maybe they just grow faster because we throw more light at them. It could just be, inherently genetic. They start to flower faster, so.
Royal Queen Seed has been putting out numbers with their auto flowering of 150 to 200 gram per square feet of dry trim flower. Are you guys seeing scores like that with yours?
I wouldn't say that's impossible. Yeah, I don't want to say too much, but I wouldn't say that's impossible.
You wouldn't say that's possible?
I wouldn't say it's impossible, but I don't want to say too much.
Oh, I was in the research room. I kind of saw. But so you're gonna get there, and okay, perfect. Any other questions out there?
Oh yeah, so for double haploid, yeah, so ploidy is very interesting, and I think Max Jones would talk about it, but he doesn't appear to be here. So normally cannabis is diploid; it has 2 full copies. Some cells are haploid, like the pollen and the egg cells. In a lot of different crops, you can grow the haploid plant through a lot of cool ways. And then what you can do is you take that haploid plant and then you apply colchicine or something, and now you have a perfectly homozygous plant derived from like a single pollen grain. And it's totally homozygous. So if you have 2 of those crossed, the offspring will be they'll be totally homogeneous. So so that's you can do haploid for these doubled haploids, diploids common, and then other people have been experimenting with higher ploidies as well to various levels of success. So if you were to create a doubled haploid, it would probably be for a parent. I wouldn't expect to see the doubled haploid plant itself to be very good. That's what they do in corn. Like the doubled haploid corn plant looks okay, but the F1 from the doubled haploids look fantastic.
The higher ploidies will often be sterile, so it won't be the feedback, right? So when you have gene dosage effects, for meiosis it doesn't work, but gene dosage effects. One of the tricky parts with cannabis is there's a lot of chromosomal rearrangements. So there's already some duplications in there. So when you then do polyploidy on top of that, the gene dosage gets really out of whack on some of those. So it's not, because there's a lot of diversity in, the pan genome was published last year. That's a lot of different genomes put together. And so they found a lot of reversions, inversions, things that are moving around already. So then when you start adding ploidy on top of that, so far from what I've heard from people who've done ploidy, they haven't really panned out the way they have in other crops yet. So it's probably finding the right combination of ploidy, and maybe it's not fully polyploid. Like corn had a big genome duplication a long time ago, but it's not the full genome. It's only half of it. And that's what went from like a single grain of corn to like a cob. So it may not even be full ploidy, it may be partial. I don't know how we get there yet in cannabis. That's a tough one. It's a tough question to answer.
One other cool thing about the sterility, especially for like triploids, if you grow them outside right next to like, a hemp field or something, they're not going to get pollinated because they're sterile. So that can be a benefit because you don't get that decrease in the potency of the flower, even if you're growing right next to a big pollen source. Maybe that, no pollination, so maybe not so great for breeding, but it can be useful for production.
I think there was a question in the back first, sorry.
Yeah, so I guess my question is about, are you seeing like, can you hear? Okay. My question is kind of in terms of outdoor cultivation at a larger scale. Do you see it going towards like the Autoflower category, just thinking in terms of multiple runs per season and also splitting your harvest, and instead of having, one big massive harvest at the end, you could potentially have two, maybe just adjusting your spacing and such. Do you see it going that direction?
I will say that is what our collaborators are doing right now. Autoflowers, they're great because you can harvest them in, August instead of October, and then you don't have it all at once, but they do yield less. And part of that is because they are, 60 days shorter in the field or something like that. So yeah, a combination of everything might be a good idea. And then you can also, we've never been successful in growing 2 cycles per year, but in theory it's probably possible. But having that mix right now is what a lot of our outdoor collaborators are doing.
Actually, I had two questions. That actually answered the first one. Second one, when you guys are getting F1 genetics from other companies, how are you verifying that you have 100% F1 genetic landrace from that? Is there any way to verify that, or
You grow them out and you complain.
You're just trusting their word on that?
So I was looking at the F1 companies out there today, and it just kind of shocked me at how many different genetics they say they have. Because
high, yeah.
Typically, in other industries, corn, they have like islands in Puerto Rico where they're breeding all year just to get one trait into an elite variety, and it takes like 5 years of just continual generations.
Yeah.
So for them to have this many different types of F1 this fast, all of a sudden there's like dozens of different companies.
Fishy.
I don't know.
Yeah.
I don't know. Because I would expect it to take 7 to 12 years to get just one inbred line.
Especially to back cross it.
And I don't know how many people were working on this that long ago. So it's interesting. I don't know.
There's no genetic test either, right?
If you had the parents. You do homozygosity tests.
Yeah.
There's sweet spots for homozygosity. If you go too far, they can be too inbred. So somewhere in the high 80s, low 90s. So there's a purebred sort of sweet spot, but you would have to have access to the parents. In the seeds themselves, I suppose you could compare. If you had the money and wanted to, you could grow them up and do some genetic testing to see heterozygosity in those and see if it matches. It should be relatively consistent among those. So it is possible. It's just, do you have the money and resources, or would you rather just grow them up and see are they different?
Yeah.
You know what I mean?
Yeah. I was just wondering, if it's based on like word of mouth, where like you're trusting the supplier who is charging a lot of money for that F1 seed, and then later you have to complain.
Yeah. And what can you do?
Like. Yeah.
No, it's a great question.
Yeah. Okay.
Normally there's specs in a contract that tells what they're supposed to do, and when you grow them out, if they're not within that, you sue the company for because the product is not what it was supposed to be.
Let's not get into that. But I appreciate the answer.
Thank you guys. We're going to have to close this panel. We have a next one coming up, so a big round of applause for our amazing panelists that came and shared the knowledge with you guys.